Bioinfocongress II Dr. Gamze Gürsoy Çoklu omik döneminde genom gizliliği. Dr. Gamze Gürsoy lisans eğitimini Boğaziçi Üniversitesi Kimya Mühendisliği bölümünde, doktora eğitimini Illinois Üniversitesi'nde Bioinformatik bölümünde tamamlamıştır. Doktora sonrası araştırmalarında Yale Üniversitesi Hesaplamalı Biyoloji ve Bioinformatik programında Dr. Mart ile birlikte genom gizliliği ve kişisel genom varyasyonu ile düzenleyici genomik arasındaki bağlantı konularında çalışmıştır. Şu anda Columbia Üniversitesi Biyomedikal Bilişim bölümünde yardımcı doçent ve New York Genom Merkezi'nde öğretim üyesi olarak görev yapmaktadır. Dr. Gürsoy'un araştırma grubu hastalıklar ve fenotiplerle ilgili olarak büyük ölçekli omik verilerini analiz etmek ve anlamak için araçların geliştirilmesi, genel sunucularda hassas genotip ve fenotiplerin analizleri, geniş paylaşım olanak sağlayan pipeline ve dosya formatları ile ilgilenmektedir. Bunların yanında özel olarak gizliliği koruyan yazılımların geliştirilmesi için çalışmalarını sürdürmektedir.
Bioinfocongress II Dr. Gamze Gürsoy Genome Privacy in the Era of Multi-Omics. Dr. Gamze Gürsoy graduated from Boğaziçi University, Department of Chemical Engineering. She completed her PhD in bioinformatics at the University of Illinois. Her postdoctoral training with Dr. Mark Gerstein, she focused on genome privacy and the link between individual genome variation and regulatory genomics at Yale University's Computational Biology and Bioinformatics program. She is currently an assistant professor in the Department of Biomedical Informatics at Columbia University and a core faculty member at the New York Genome Center. Dr. Gürsoy's research group is interested in developing tools to analyze and understand large-scale omics data in relation to diseases and phenotypes, with a particular interest in developing privacy-preserving software, file formats, and pipelines that enable broad sharing and analysis of sensitive genotyping and phenotyping data in public servers.
Sayın Dr. Gamze Gürsoy ve değerli katılımcılarımız, Bioinfocongress II etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün Genome Privacy in the Era of Multi-Omics konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüz olacaktır. Merhaba Gamze Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkür ederim davetiniz için.
Biz teşekkür ederiz kabul ettiğiniz için. Dilerseniz sunumunuza başlayabilirsiniz.
Evet, bunu tekrar deneyelim. İronik bir şekilde privacy ayarlarına takıldık. Çok özür diliyorum geç kaldığım için. Şu an bütün slaytları görebiliyor musunuz?
Şu an göremiyoruz hocam.
Tekrar. Aha, evet.
Evet, şu an görebiliyoruz. Tam ekran yaparsanız.
Şimdi tam ekran oldu mu?
Evet hocam, gayet iyi.
Tamam. Çok teşekkür ediyorum. Buradan sonrasını İngilizce yapacağım. Umarım sorun olmaz. It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago. And I'm really excited to share with you what I've been doing in the past 4 or 5 years in my postdoc. And just a little overview, as I was trained as a computational biologist and bioinformatician, I'm interested in many aspects of biological data, but today I will heavily talk about my genome privacy work. slide geçişini görebiliyor musunuz? Gördüğünüzü assume ediyorum. Today, I'm going to first start with explaining a little bit basics of paradigms in science, and then you will see why I'm talking about this. As you all know, the paradigms in science starts with the empirical analysis, theoretical background, and the computational sciences. But here, by computational sciences, we mainly mean the analysis, not data analysis, but numeric methods or other computational ways of using the theoretical data. But with the increasing use of data, there is now a paradigm shift to the nature of science toward data science. And we were actually indeed warned about the flood of data that we have to develop new tools to cope with it. This is particularly true in the era of personalized medicine, where we are collecting a large amount of data from patients and research participants. And the ongoing work by the, all over the world with the informatics faculty, that we are getting better and better at dealing with the challenges that the data-intensive discoveries present to us. But there are some efforts addressing the challenges related to privacy and security of the collected data that is still at its infancy. Let's think about how we use genomics, for example. In order to understand a genetic disease, we need to make connections between the genomes and the disease phenotypes. But unfortunately, direct linkages do not really exist because the effect of genetics is propagated through different pathways and biological mechanisms. That's why we need data from other genomic-related sources, such as personal epigenomes, transcriptomes, proteomes, and metabolomes. And this creates issues related to analysis, sharing, and the privacy of the collected data. I feel really lucky to be doing genomics in this really exciting times where we collect and have access to many different types of genomic and related data. This information explosion comes with an analysis gap that calls for transitioning the analysis from traditional approaches to data-driven approaches, such as machine learning. Because as I mentioned, the relationship between the outcomes and the input are complicated to capture beyond simple statistical methods. However, one of the barriers in increasing the amount of data to be used in machine learning for better statistical association is the private nature of this data. Therefore, the overarching goal of my research during my postdoc and also as well as in my new lab is to address the challenges of privacy, analysis, and sharing of personal biological data in an ethical manner on our way from data production to personalized medicine. Today, I structured my talk to answer these two questions. How we can quantify the genetic information leakage in omics data, and how we can develop privacy-preserving sharing and analysis of omics data. So I will first start with genetic privacy. So let's talk about what genetic privacy is. As you all know, our genomes are our fingerprints. We share 99% of our genomes with the humankind, but that 1% difference is what makes us different from everyone. It's extremely unique. It can tell roughly which geography we are from. It can tell what kind of health problems or traits we will have. I think by now I don't need to convince people that removing identifiers such as names, date of birth, or zip codes will not de-identify the genetic data. And indeed, it was shown that in a very elegant way that we can infer people's last names or full identities by using genomes. DNA data leakage is also more dangerous than other data leakages because you cannot change your DNA. You can change your credit card if it gets stolen, but if your DNA data is stolen, there is no change. And also, it does not only affect you, but affects the generations after you. And here, the tip of the iceberg is typically what you see in the media about genome privacy. Things like how the current laws does not cover life insurance, or how police can knock on your doors because your relative might be a criminal, or how 23andMe is allowing drug companies to have access to their customers' private genetic data. But there are also other aspects of genetic privacy stemming from the dark history of genetics, especially in the United States. Easy access to genetic data could lead to things like discrimination through racial profiling. There's also the issue related to data ownership, inclusion, and who benefits from data sharing. There are communities here who are really concerned about these issues. Of course, as scientists, we should not be extremely alarmist and cause situations like our access to data will be difficult or completely cut, because we can only advance biomedical science if we have access to data. But we must be prepared to create infrastructures, technological advances such as privacy-preserving technologies, and better policies to create trust between people and the researchers. This will only increase the participation to the studies, and that will enable better statistical power for more medically and biologically actionable discoveries. So the leakage from the genomics data is kind of easy to understand. If you share your entire, 3 billion base pair letters of a person's genome, you can mine these letters and find what is different between this person and the others and use it for all sorts of inferences. But now, as I mentioned, there are many other types of molecular measurements that are similar to genomics. For example, transcriptomics data tells you which genes are expressed and which genes are silent. Epigenetic data tells you what are the factors and the proteins that bind to the DNA. And we call all of these measurements as functional genomics data. The question is how we can determine the private information leakage in these other types of omics data. Let's talk about RNA-seq as an example. Let's say we have an individual with a phenotype. We want to understand which genes are up and down regulated in this person's genome. After we sequence the RNA and overlap these sequences with the locations of the genes in the genome, and we can conclude which gene is expressed and which gene is not expressed. So at the end, what we want is this gene expression matrix, but it comes with these next generation sequencing reads that contain the genetic variants of the individual that the sample is taken from. And therefore, following traditional DNA sequencing data sharing protocols, we store this raw functional genomics data in restricted data archives such as dbGaP. And this creates bureaucratic problems, especially for people who are accessing the data outside of the United States, because there's a huge access problem to the data. But if the data is legally de-identified, what can be learned from just a collection of these letters? I want to show you that even after we noise this data significantly, we can still use these genetic variants as keys to open up more sensitive information about these individuals. And actually, this quantification allows us creating solutions to this privacy problem, and hopefully we can share this data more broadly with the researchers. One way to reveal this amount of privacy leak is to perform a linking attack to the data. Linking attack is typically a privacy breach where we overlap two data sets in order to learn sensitive information about an individual in these data sets. Here we have an RNA-seq data from a cohort of approximately 500 individuals, and let's assume half of them have the bipolar disorder positive and half of them have the bipolar disorder negative phenotype. We also say steal someone's genome, like our friend Jane's genome, and want to know if Jane is in this RNA-seq cohort and if she has bipolar disorder or not. So what we do is we first obtain genotypes from RNA-seq reads. These genotypes are not accurate, they are partial, as RNA-seq cannot capture the entirety of the genome, and you can see from the precision and the sensitivity of these genotypes, they are quite noisy. And we already have Jane's genotypes from the stolen genome. We can then intersect Jane's genotypes with every single anonymized individual in this cohort and score this intersection based on the allele frequencies of these genotypes, meaning that if there is a rare allele in Jane's genome and a rare allele in the RNA-seq data, that means that that genome in the RNA-seq data might be belonging to Jane. We can then rank these scores based on how good they are, and then we can conclude that the top-ranked individual is Jane in this anonymized cohort. And to have the statistical significance and reliability, we can define a metric called gap that tells you how different the top-ranked individual than the rest of the cohort. And if this gap compared to a randomly distributed gaps is significant, then we can reliably say Jane is the 10th individual in this first cohort, meaning that Jane has bipolar disorder. So this is great, it works for Jane, but how accurate is it actually? We did this for every single individual in this 500 people cohort and successfully and significantly linked all of them back to their RNA-seq data with 100% accuracy. But this is somehow an easy linking attack because we stole someone's genome. With the current protections in place, it is really unlikely that we can steal genomes that easily. So for that reason, we assessed if we can still link known individuals back to their RNA-seq data when we have noisy genomes from the individuals. And one way to collect noisy genomes is to use discarded items such as coffee cups. It's also perfectly legal to grab someone's coffee cup and sequence it. So what I did is I first collected blood from consented individuals, extracted the RNA, sequenced it, and added to my anonymized database. So I have now added this person's RNA-seq as well to that bipolar disorder cohort. And then I collected used coffee cups from the same individuals, extracted DNA on the surface of the lid and sequenced it, and then performed a linking attack between the coffee cup DNA and the RNA-seq database. And this was performed for 2 individuals with 6 coffee cups from each. So when I did that, first I want to show how noisy these data sets are, and then I will show you how good we are in linking the coffee cups back to the RNA-seq data. So I already mentioned that the RNA-seq genotypes are neither precise nor sensitive. The genotypes from coffee cups have also very low sensitivity because we cannot obtain the entire genome. You really need to torture the DNA on the lid to be able to get something out of it. There is also a lot of contamination. We have a lot of false positives because, there is the environmental DNA that it is contaminated with. There is also, if you're buying the coffee from a coffee shop, the barista's DNA is on it too. But regardless, when we sequenced the coffee cup DNA at 10x coverage, we were able to link all 12 of them back to their owners in the RNA-seq database. We then downsampled the coffee cup DNA and found that even at 0.125x coverage, we are more than 60% accurate. This is around $19 worth of sequencing. So with just this includes all the reagents that I use, meaning that if I just grab someone's coffee cup and spend $19, and if I know if I have an anonymized RNA-seq database, I can link that person back to their RNA-seq database. So with this and also with the previous linking attack, we were able to tell the degree of the leakage and understood that we should not be really treating this data saying that, oh, this data is really noisy, we shouldn't be, we can share it broadly. There is actually a really big privacy leakage in these RNA-seq data and also other functional genomics data such as ChIP-seq and ATAC-seq as well. So let's go back to the raw data. We really want to share it, but currently it is stored in the control access databases, and this reduces the utility of the data by creating bureaucratic hurdles to the researchers. And these hurdles are even a lot higher for researchers from outside the United States. But it provides privacy. We cannot share it publicly, as I mentioned, that there is a huge privacy problem, but it will have utility. So we need to find a solution that balances the utility and the privacy needs. And this is kind of possible for functional genomics data because we know that genetic variants are the byproducts, and we often don't need them to do most of our calculations. So, for example, a lot of these rules, sharing functional genomics data behind firewall were established during the DNA sequencing and genotyping was popular. With the increasing technology, we have now more and more omics data being generated, and all of this data is being treated as DNA sequencing in terms of privacy and sharing. There are two main problems with that. First, the penetrating privacy is easier because we have now more information, as I showed. But also now we will have this overflow in these central data storage repositories, because you can see that the amount of the data you get from functional genomics is a lot larger than the DNA sequencing data, because for whole genome sequencing, you sequence someone just once and you have the genomes. For functional genomics, you can perform functional genomics to many different tissues of the same person, or you can do different kinds of functional genomics such as ChIP-seq, RNA-seq, or ATAC-seq to the same person. So meaning that we have more data that we need to liberate from the firewall, but we also have more privacy problems. So we thought that since we don't use genetic variants for calculating, for example, gene expression values or ChIP-seq peaks, there should be a way to sanitize this data and share it more openly. Before diving into how we can do that mathematically, I want to explicitly show you the source of genetic variants and introduce you commonly used file formats, which I know that most of you already know these, but it's helpful to go over the basics. So when we sequence RNA, we obtain reads that are of particular length, and they are the combination of letters in our DNA. And this data is commonly stored in a format of FASTQ files. We then take every read in these FASTQ files and map them to the coordinates of human genome and derive another file type called BAM files. These store the locations which these sequences are coming from in the reference genome. So meaning that you can actually take a sequence from an RNA-seq BAM file, look at the locations in the reference genome, and then figure out what the difference between this person's genome and the reference genome. And this is called variant calling, as you know. But it's really important to share the files in the form of BAM files because they don't only preserve the data in a compressed format, but they are also used as input to many functional genomics data processing pipelines. So it's important to make the data available in BAM format, but also important that we provide privacy. Currently, we store the functional genomics raw files behind a firewall where we keep it from the bad actors, but it also keeps the data away from the scientists. In the next few slides, I want to explain ways to sanitize the data such that the private components of the data can be kept away from the bad actors while we give the public component to the scientists so that they can do easily their calculations without going through bureaucratic hurdles. For that, we developed a privacy-preserving transformation that takes the BAM files and transforms it to a privacy-preserving BAM, PBAM in short. This is done by making sure that any calculation F that is done on the original BAM file and the PBAM file will yield very similar results, and the difference between these results should be quantifiable a priori. We do this by replacing the genetic variants with reference alleles, although it is really straightforward to do this with SNPs because you just flip the the base pair. If there's a SNP, you convert it to the reference allele. But it's not as straightforward for SVs, structural variants, because you have these big gaps or big insertions in the data that you need to make sure that when you do this sanitization, it doesn't hurt the utility of the file. So we can actually mathematically define the privacy and utility. Privacy is defined as the percentage of the observable variants that are masked, and the utility is defined the percentage of the genome that is above an error threshold, meaning that when we do a transformation to a structural variant, we can actually count how many locations in the genome we are noising when we do that, and then using that number, we can actually calculate the utility. And now we can also show that there is a mathematical balance between the utility and the privacy by deriving these equations. And we can numerically show the upper bound of the utility loss when we have 100% privacy. So this is the absolute maximum utility loss that you can get from any functional genomics PBAM file. And we are showing that it is at most 10%. And in reality, actually, it's a lot lower than that. So for example, here, if you look at the difference between gene expression between BAM and PBAM in blue, it's very consistent. And you can also compare it to the difference between the first replica and second replica of the same sample, that you can measure it within the context of the biological noise. And you can see that the variation between BAM and PBAM is actually a lot lower than variation between the replicates at 100% privacy. Here I want to show how it plays out with privacy. In this top figure, I am increasing the number of genetic variants that we sanitize in the x-axis, and then in the y-axis, I am calculating the risk of re-identification of this person, which is, as I showed, that it is calculated by this metric gap. And if this gap falls below this threshold, which is the dashed line, then I know that I can no longer re-identify this individual. And then here in the blue line, I am showing how much noise I am adding to the data by doing the data sanitization. As you can see, there is this sweet spot where you can no longer identify the individual, and you also add a very small amount of noise. We can also define privacy in terms of the number of GWAS SNPs observed, because if you're observing a GWAS SNP about in an individual's genome, you can pretty much tell a phenotype about that individual. For example, if this person has this diabetes risk genetic variant, then you can more or less say that this person has either diabetes or has the risk of diabetes. So as we increase the sanitization of our data, we remove more and more GWAS variants. And then in the blue, I am showing how much error we are adding to the data. And again, there is this similar spot where you don't add much error while you actually provide a lot of privacy. So in summary, with this data sanitization, a large amount of privacy leakage can be prevented, and the data can be shared broadly. It works with all usual suspects, from ChIP-seq to RNA-seq to ATAC-seq. We also developed these tools called P-tools, where we can do the sanitization, but also what we do is we create these small files called Diff Files. These files has the information, the difference between the PBAM and the BAM. And you can put these Diff files behind a firewall. So the idea is that a researcher can access the PBAM freely. And then if the calculations are useful for that individual, for that scientist, then the scientist can go and apply for the Diff Files, so we can actually cut on these bureaucratic hurdles a little bit. And also Diff Files are a lot smaller than the BAM files, so it's easier to keep them behind a firewall. And with P-tools, you don't actually ever have to store the BAM files. You can take the Diff Files and the PBAM and the reference genome and then go recreate the BAM files. So here I argued that the policies to protect privacy of raw functional genomics data is too stringent, and there are ways to sanitize this data and make this data freely available. And then next, I want to argue that policies to protect privacy in summary level omics data is too lenient, and we need to do something about it. So let's talk about summary level data. It has been repeatedly shown that you can indeed infer genetic variants from summary level omics data. So here I am showing raw reads, signal profiles, and summary results, which is the gene expression in the case of RNA-seq. As I shown that raw reads have problems, and it has a lot of variants, genetic variants that you can infer. And it was also shown that signal profiles leak deletions. And it was also shown that gene expression values can correlate with eQTLs, and you can actually figure out people's SNPs by looking at the gene expression values. And here we are showing that how the amount of data, decreases as you summarize the data, but also privacy leakage also decreases, but then you get less utility as you summarize the data. But now I want to talk about a specific type of summary level data that leaks privacy that was not thought of before, which is allele-specific activity. So what is allele-specific activity? Let's talk about expression for now, for simplicity. So allele-specific expression is becoming more and more clinically relevant because it helps to understand the effect of genetic variants without the environmental factors, because you look at a single individual's genome and compare one allele to other, maternal genome to paternal genome. So let me quickly refresh your memory on allele-specific expression. As you know, we have two copies of chromosomes, maternal and paternal. Sometimes we have these letter differences, which are the heterozygous SNPs on our genome. When we calculate the gene expression of a gene from RNA-seq, it is the total expression that comes from the both copies. But by looking at these letter differences, we can actually deconvolute all the reads that we have into maternal and paternal copies, and now look for the gene expression in different alleles. And this is called allele-specific expression, or allelic imbalance. So because we have, by looking at the reads, if the read contains A, it goes to the maternal. If the read contains C, it goes to the paternal. So what can we learn from allele-specific expression in terms of privacy? So the question is, if we have a list of allele-specific genes for a donor or a patient, can we somehow show or predict a list of genetic variants for that individual to be used in a genome privacy attack? So here's the idea. To be able to identify a gene having an allele-specific expression, the gene has to have a heterozygous SNP on it or around it. Because without the heterozygous SNP, you cannot tell if the gene has allele-specific expression or not. So we can use this knowledge to penetrate privacy. For each gene, we can overlap the locations of the exons with the heterozygous SNP locations in a population of individuals. And then if the SNPs are not rare or not more common, we can then assume that this individual might have these SNPs, and because of that SNPs, we can tell if the gene is expressed in an allele-specific manner. So we can actually create these potential SNPs for that individual. And what can we do with these potential SNPs? In the first scenario, if we have an anonymized individual with these potential SNPs, we can attack to a de-anonymized database, such as GEDmatch, where we know the individuals and we know the genotypes. And then for these known individuals, we can predict phenotypes, because the likely that anonymized allele-specific expression list will come with a phenotype. If we know the person and we have these potential SNPs, I don't know, maybe we get access to it somehow, we can attack to an anonymized genomic database, and we can find the full genome of this person by using these potential SNPs. And how accurate is this? Because we are just guessing SNPs. We don't know exactly if this person has those SNPs or not. But surprisingly, we found that we are able to identify around 55 to 60% of 300 individuals that we did this attack for. And then we tried different versions of this attack. For example, HLA genes are allele specific in many, many individuals, so they don't contain really useful information because you get the same SNP list for every individual. So if you use just the HLA genes, we see that we cannot actually identify anyone. I mean, around 1%, I think. If we remove the HLA genes from the list of the genes, we can actually increase our identifiability by 5% or so. And if we actually remove all the common genes that we see in these allele-specific gene lists from the patients, we can actually even more increase and go up to 70% of accuracy. And this is a lot of accuracy, actually, because if you think of it, we are identifying 1% out of 3,000, meaning that our random chance is 1 in 3,000. So 70% is pretty high. So with this, in summary, a list of allele-specific genes without any explicit genetic variant information was enough to re-identify a large number of individuals. But by no means that we suggest that we should not share this kind of summary-level data because it's very useful for science. But we think that this knowledge should be incorporated at the informed consent processes, at the education of the participants, as well as for future lawmaking. So why are we constantly restricting access to the data? Right, because we keep finding these privacy issues and we are constantly restricting access. The problem is that there is this knowledge gap. And if there are any computer scientists around you, you might know that there is this disconnection between these two fields where there is the biologists who are generating these vast amount of personal biological data and analyze them. And then there is the fields of computer scientists and they set the theories behind privacy and security. And with the changing policies and laws, it's becoming extremely difficult to access the data in the plain form. So our mission is to merge these two distinct disciplines and make better statistical associations between genomes and the diseases using cryptographic techniques such that we can also combine data from multiple different sources in a privacy-preserving manner. So having said that, now I want to talk about how this technology, the cryptography, applied cryptography, can help with a lot of these privacy problems. Before diving into how we utilize cryptography for DNA sequencing data, let's talk a little bit data privacy in general, because this is not news. Data comes in three forms. Data in transit is protected through encrypted connections. We encrypt the data in rest, but if we want to use the data, we have to decrypt it. Protection of data in use, that is we are interested in, is especially in cloud services, is a difficult problem to solve. There are several solutions proposed, such as trusted executive environments, secure multi-party computation, or homomorphic encryption. But they are really, they are very nice because they have theoretical guarantees, but they have huge challenges related to data performance, applicability, and usability. Especially when it comes to genomics, the overhead from the amount of data and also from the many nonlinear operations that we do, these challenges becomes huge. These technologies allow us to work on the data while protecting the data at the same time. So meaning that you can put the data in the cloud in an encrypted form and work on the data while the data is still encrypted. And we can think of it as, a jeweler working on a piece of very expensive and important piece without taking it out of this safe box. So how does this work? Let's focus on a particular problem because every different problem in bioinformatics will have a different form of solution when we use applied cryptography. Today, I want to talk about particularly this problem, genotype imputation, because it's really one of the applications that has been done using homomorphic encryption that we can now scale it for a lot of genomes. So this is a famous graph from NIH where you see that there is a steep decline in the cost of human genome sequencing for a single genome. So but we want to have statistical power, meaning that we want to sequence thousands of genomes, and it's still very expensive. So we still do these, traditional ways of looking at the genetic variants, such as gene chip approach, where we assay only a subset of the genetic variants in the genome, and then we predict the rest by using the correlations between the different genetic variants using for example, linkage disequilibrium. For example, here I'm showing that these pink circles are the target SNPs where we get from the gene chip. For example, if you do 23andMe, what you get is a subset of your genome, and now you can predict the rest of your genome using these target SNP genotypes. So here I'm going to show you how we can perform this analysis using homomorphic encryption. And the idea is like this. Let's say Bob is a cloud provider, and he has an imputation model that works with encryption. Alice has her VCF from 23andMe, and she wants to impute the rest of her genome, but she doesn't want Bob or the cloud server to see her genome. So what she does is she encrypts her genome, sends it to Bob. Bob imputes the rest of the genome and outputs an encrypted file and gives it back to Alice, and now Alice can decrypt it and use it. So this is developed using a technique called homomorphic encryption that allows this computation to happen on the encrypted genome. And how does this work? So I'm going to dive into the details of this algorithm a little bit. So first, I'm going to talk about how I came up with a very simple plain text solution, and then I will tell you how I convert it to homomorphic encryption. So let's say this is the genome, and these pink circles are the genotypes that we get from the gene chip analysis. For example, your 23andMe results. And these, the other way, the purple ones are the ones that you get, and the pink ones are the ones that you want to predict. So for each target SNP that we want to predict the genotypes for, we can define a window and number of tag SNP around it, and we call it a pseudo haploblock. It's not a real haploblock, but it's just a guess of what the haploblock around this target SNP might be. So what we can do is, for this individual, we know the purples, we don't know the pink. We can go, for this particular region, go to our database that we have full genomes for example, 1000 genomes database, and we can look for individuals that are similar to our query individual with respect to their purple circles. So we have a way of calculating this similarity, and then we can rank all the individuals in the database based on the similarity to the query individual, and then take the top M individuals. And then we say that this top M individual is the genetically related individuals to this query individual for this pseudo haploblock. And then by using the joint probabilities between the pinks and the purples, we can actually calculate the probability of this target SNP genotype being 0, 1, or 2. So this is great, but the problem is that when we calculate this, every time we have to go to the database and do this similarity calculation, and it's very expensive, and we cannot do it with homomorphic encryption because homomorphic encryption is designed to only do simple operations such as summations and multiplications. So how can we convert this into a version where we can do it with encryption? So for this, what we did is, let's say we assume that, so we know that in the pseudo haploblock, all of these tag SNP genotypes, the genotypes that we know, can only take three values, right? Either there is no SNP, or there is SNP in one allele, or there is SNP in both alleles. We can actually simulate all possible individuals, enumerate all possible individuals for this pseudo haploblock. And then for all these virtual individuals we call possible individuals, we can actually calculate what the pink circle will be for each of these individuals, and then create a lookup table. In this lookup table, our index is the sequence of the tag SNP genotypes, the sequence that we know. And then the second and third column are the probability of the pink circle being 0 or 1. So now we pre-computed for every single individual on Earth what this pink circle will be. So we can then encrypt this lookup table. So when we have a query genome, we know that that genome exists in this table because we calculated for every possible individual. We can easily look up for what this query genome's pink circle will be. Either it's 0, 1, or 2, meaning that the probability of 2 will be the 1 minus probability of 1 plus probability of 0. And this is called a private information retrieval problem. In plain text, it's very quick. It's a lookup table. In encrypted domain, it scales with the number of rows, but our number of rows is actually bounded by 3 to the power of n, meaning the number of tag snips. We use this encryption scheme called VFV using Microsoft Seal's library, and doing some tricks such as batching and no bootstrapping, we make it really fast, and we can now, in the encrypted domain, calculate the genotype imputation very easily. And here I'm showing and comparing the results of P-Impute, which is our program, how it compares to the state-of-the-art plain text solutions, Beagle and Impute2, that are the most commonly used imputation solutions. So we can provide privacy, and we are almost as good as Beagle and Impute2. There is a loss of accuracy with there's a trade-off between the privacy and accuracy, but it's still quite accurate. And this is very scalable. We can actually calculate genotype. We can do genotype imputation for 1,000 of individuals in 8 minutes by using this encrypted way. So basically, this is a phasing-free, scalable, and privacy-preserving and accurate genotype imputation method. We use fully homomorphic encryption. It enabled us outsourcing this computation on genome from large cohort to cloud services. And encryption has its problems because now we encrypted all this data. The size of the ciphertext increased exponentially with the increasing data size. And also, as I mentioned, that this solution is specifically tailored towards genotype imputation. For every bioinformatics problem, we have to create a different kind of approximation such as this. So now I'm going to talk about, the genotype imputation is actually an inference problem, right? We have a model, we have an observation, which is the tag SNP genotypes, and we want to know what the outcome is, which is the target SNP genotypes. There are also privacy issues when we do machine learning training, because we are using observations and outcomes from a large cohort of patients, and models also has problems because model can leak who is in the training data. So since I talk about the inference problem, I want to talk about just very quickly how we can solve the privacy issues in machine learning training. And I'm going to talk about this this framework called Federated Learning and differential privacy, and how it can help us to do machine learning in private settings. So let's say we want to predict whether a patient has breast cancer or not using their gene expression values. However, we do not have enough number of patients in a single hospital to train a model with good predictive value. So if we combine the number of patients in 3 different hospitals, then we reach enough statistical power to have a good machine learning training. But due to the privacy policies of these institutions, it's not possible to combine these data in a single server to perform the machine learning training. So then we can perform federated learning, meaning that we train the models independently in different institutions and combine the models in a third-party server. This will also have some privacy issues because model, as I mentioned, might leak information about the training data in different hospitals. And for that, what we can do is whatever we are sharing with the third-party server, meaning the model parameters, we can add a little bit noise, conforming with the differential privacy. And then based on our privacy metrics, we can actually combine the noise models. And we do this until our model converges to a good model, meaning that every institution have the same model parameters. So we created a mock network of hospitals with the data from TCGA to perform this training and evaluated our final federated model in an independent test set. We used a simple neural network model that are composed of 3 fully connected layers, where we used a ReLU function and a sigmoid for the final classification task. Our input is the gene expression values of all protein coding genes, and our output is the probability of this patient having breast cancer or not. To make this training more realistic, we focused on 4 different scenarios. In the first scenario, we have equal number of patients in all 3 hospitals, and also in each hospitals we have equal number of BRCA patients and no BRCA patients, meaning breast cancer patients versus no breast cancer patients. In the second scenario, we have again equal number of patients in each site, but the proportion of the sick patients to normal patients are different. In the third setting, we have different number of patients in different hospitals, but the proportion to the sick to normal patients are equal. And in the fourth setting, which is probably the most realistic setting that we have, it's a non-IID setting, meaning that we have different number of patients and also different number of ratios between the sick and normal patients. So here in this plot, what I am showing is on the y-axis, it's the accuracy of our machine learning training on an independent test set. And then on the x-axis is the privacy level. If the epsilon is small, meaning that there is a lot of privacy added to the system, and if epsilon is large, then the privacy, we don't have as much privacy. So what I'm trying to show here is that even at very stringent privacy levels, in a non-IID setting, we see that with our framework, we can provide really high accuracy. So this is really promising, but it's again tailored towards this particular problem. But this is promising in a sense that we can have this privacy-preserving machine learning training where we can train thousands of patients that are in different hospitals. And this specifically is important for perhaps rare disease discoveries where we don't have many patients in single hospital, but now with this, we can create these federated networks and maybe come up with some algorithms where we can predict if different types of things, for example, rehospitalization or different types of treatments, if the treatments will work for these patients in a federated setting. So here are my 3 takeaways for you for my presentation. In order to understand the relationship between the genomes and the diseases, an enormous number of measurements need to be made. And this data is really enriched with beautiful biology, but it comes with a price, which is privacy. The privacy-preserving analysis approach is now technologically feasible and can be an important novel focus in human genome research. So I highly encourage, if there are any of you, trying to pursue academic research, this could be a really nice side track kind of in human genome research, but it really is useful and almost translational because you provide a lot of infrastructure to the researchers to do better predictions and better disease models. With this, I want to thank my newly minted lab members for taking a chance on a new PI and joining my lab as soon as I started. We're still hiring, so if there are any interested graduate students, please don't hesitate to contact me. Our hiring for grad school is done through the school, but if you can reach out to me, I can provide you what you need to know, how to apply. And if you're looking for a postdoc or a staff scientist position, please don't hesitate to contact me. I can't promise, but there's a due process, but I will definitely respond back to you based on your CV. We also love collaboration, so please reach out to me if we can be of any help with the scientific questions you're attacking. The vast majority of the work I have described here was done during my postdoc at Mark Gerstein's lab. So I'm really grateful for his guidance and all the members of the Gerstein lab, and also my PhD lab in Jay Liang's lab at University of Illinois. And I have a lot of great collaborators through ENCODE Consortium as well as at Yale. And all the privacy-preserving homomorphic encryption work is done in collaboration with Mihalis, who is at NYU. With this, I'm of course very grateful of Columbia University and New York Genome Center for the support. I was hired as part of Columbia Precision Medicine Initiative, and tremendous thanks to NHGRI who supported my vision. And with this, I want to thank all of you for having me here today, and I would be happy to take any questions.
Çok teşekkür ederiz hocam. Dilerseniz ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüze geçebiliriz.
Tabii ki.
İlk sorumu soruyorum. MapQ scores of different bioinformatic tools like BOV-TAE2, BIVA-MEM, and TopHat vary significantly. What is the reason behind it? Which one is more accurate to use?
This is a great question to ask to developers of BOV-TAE and MEM or TopHat. I have no idea why they have different scores. So, I mean, MapQ scores in general just tells you how good the mapping quality of a base to the reference genome. I think that there is correlation between all these scores. So if you plot, for example, MapQ in BOV-TAE2 versus BWA-MEM, you will get high correlation. It's just maybe scaling is different. And I have no idea why they have different scoring systems. Sorry.
Cevabınız için teşekkür ederiz hocam. Sıradaki soruyla devam ediyoruz. What is the place of blockchain in genomic storage?
Another great question. Thank you for asking, whoever asked this. Well, so this is a highly debated topic. The blockchain can be extremely inefficient to store large-scale data. So even though it creates really this nice infrastructure with, cryptographically, how to say, it has this immutability and security properties. So it is actually a really great infrastructure to store really sensitive data, but in the meantime, it is very, very inefficient. So I think as the technology advances, we will have better, network systems with low latency, and maybe we will also come up with better consensus mechanisms in blockchains that makes it faster. And then maybe there will be a place for genomics. For now, it's in its infancy. I did some proof of concept work. Not as applicable as real life problems yet, but I think we are getting there.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz YouTube katılımcımızdan geliyor. What kind of applications can be made so that the information that they can access our genetic code from the products we use in chain stores does not terrify people?
So as far as I understand, the question is about how we can share the data, perhaps in blockchain, so that it's private, so that the people are not worried about sharing their data. So in for example, in private blockchain networks, you can give selective access to people. So for example, you have your genome, you store your genome in a blockchain, you own the network, and you can choose and pick and choose who you want to give access. And maybe that's one way. So the traditional way is that you give your genome to the researcher and they are in charge of your data, or you give your genome to 23andMe and they are in charge of the data. And I think a better model is that when the individuals are themselves in charge of their data, then that way they can actually give selective access to other researchers or clinicians. And I hope this answers your question.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz. COVID-19 için alınan swab örneklerimiz araştırılmaya açık genetik bankalarına gidiyor olabilir mi?
Very good question again. I think if that's the case, they have to provide it in the informed consent. So when you sign the consent to give your data, you have to be very careful to see if it's going to a biobank to do more research on it. And also it depends on how they are isolating the information from the swab. So if it's just the COVID, then they are taking the virus RNA, doing PCR on it, and maybe discarding the rest of the sample. But the sample will contain your DNA. And if in the informed consent they mention that, then they might be doing that. But it's illegal to do that without letting you know. So if you're signing a piece of paper, please look into that.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz. Tohum verimliliği arttırmada tek nükleotip polimorfizm bağlantılarının yeri nedir?
I unfortunately am not very familiar with, this kind of agricultural genetics field. So I cannot really, I don't feel qualified to answer this question. I'm sorry.
Teşekkür ederiz hocam. Bir sonraki sorumuz. Cases of unauthorized use of genetic information result in rejection of participants against ethically dissent studies. How can people's perspective on this issue be changed?
So as far as I understand, Zeynep is asking, because of the unauthorized access to the genetic information, people might be reluctant to give their genetic material to the study, result in rejection. So if that's the question, I think the best way to do is to provide privacy-preserving infrastructures and then make sure that the patients and the participants know that the data will be collected and shared in an ethical manner. And putting the trust on individuals, meaning the scientists, is one thing, but putting the trust on a mathematically guaranteed software is another thing. So I think the algorithms and technology can help in that.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz. Tek nükleotit polimorfizmde kullanılan veriler CRISPR ile düzenlenerek tasarlanmış bir in vitro fertilizasyon yapılabilir mi?
Again, I don't feel qualified to answer this question, but it's a great question. And yeah, I don't know. I mean, the CRISPR is also another field that is, really taking off very, very quickly.
Teşekkür ederiz hocam. Bir sonraki sorumuz YouTube katılımcımızdan geliyor. Because the tools and methods are largely isolated, can a uniform framework be obtained that can effectively process and analyze multi-omics data end-to-end, along with easy and biologist-friendly visualization and preparation? Çok özür dilerim hocam.
Rica ederim. This is a great question. That's what we are working for, all the bioinformaticists. We're trying to uniform frameworks from end to end, and also I work on the privacy preservation of that, so that everything is really easy for the biologists to do. But as was mentioned, the data is, the data tells you different data tells you different stories, and also different problems have different performance issues. This is the goal, but when can we get there? I don't know. We are working on it, but hopefully, in the future, we will have these frameworks, one-click frameworks in clouds where biologists goes, they input their data and push a button and everything is analyzed. And I think that there are now infrastructures to do that, but it's I'm not sure how uniform they are, but yeah, this is the goal.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz. Does the use of gene chips have advantages over the use of microarrays?
I think they are the same thing, though. I mean, it depends on what you mean by microarrays. If gene expression, microarrays do gene expression, gene chips do SNP calling, finding the SNPs. Yeah, I don't know if this answers your question.
Arkadaşımızdan soruyu daha açıklayıcı biçimde yazmasını rica edebiliriz. Daha sonra bakabiliriz isterseniz hocam soruya tekrar. Sıradaki soruya geçiyorum. Multi-omik kaynaklar hastalıkların önlenmesi veya tedavisi bakımından tıbba nasıl bir katkı sağlamaktadır?
So with multi-omics, you can assay many different properties of the genome. For example, if you have a disease that are caused by a gene, but the gene itself for that patient doesn't have a SNP on it, meaning there is no genetic variation on that gene. So you're trying to figure out what is the genetic variation that is causing the disease. So what you can do is you can look at the other multi-omics data that tells you what is bound on that gene, for example, a transcription factor, and perhaps that the gene that is encoding for the transcription factor is having the genetic variant. Or you can look at, for example, high-C data that tells you what is the interactions between that gene and the non-coding genome that might have the genetic variant that are causing the disease. So it helps you to annotate all these non-coding or coding genetic variants that are showing up as high, they are showing up as significant in GWAS, but you don't know what they are doing. So the multi-omics allows you to do that because it allows you to annotate all the non-coding genome, not just the genes.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz. Lots of genomic analyses rely on the genomic variation between individuals. As far as I understand, you mask these regions in PBAM files.
Great question.
So I imagine there must be some type of analysis that are not suitable for PBAM files. Is there such a thing? What are some analyses that are suitable and unsuitable for PBAM files?
Yeah, this is a great question that I get all the time. PBAMs are designed for functional genomics data, meaning that you can calculate gene expression, you can do peak calling, you can do, chromatin accessibility analysis, where are the regions that are accessible, where are not, but you cannot do genotyping, which you shouldn't be doing genotyping on functional genomics data because you have whole genome sequencing data that you can get more uniform information. you cannot calculate the allele-specific expression or allele-specific activity because you have to know the genetic variant. So there are use cases for PBAM, and then if the use cases are outside of these functionalities, then we highly encourage people to apply for the Diff Files, which are smaller than the BAM files, and then do this analysis.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuzla devam ediyorum. Are these single nucleotide polymorphism can be benefit to explain evolution?
Again, I am not an evolutionary biologist, so I will also skip this question saying that I'm not qualified to answer.
Teşekkür ederiz hocam. Bir sonraki sorumuz YouTube katılımcımızdan geliyor. Depending upon the transcriptomic data reported about any particular disease, is it possible to find marker for that gene with that data? How much it will be reliable?
So I'm not sure what by marker, I'm guessing it's SNP for that gene with that data in the transcriptome. Yeah, so in RNA-seq you can find these variants that are on the genes. How much it will be reliable is I showed that the RNA-seq is very noisy. So the SNPs that you get from RNA-seq, these markers, might be noise due to post-transcriptional changes or due to how the data is processed. But there are now tools that can be really accurate SNP calling from RNA-seq. So if you use those, it would be reliable. What I did is very quick and dirty SNP calling because I was doing it for privacy reasons, not for actually finding the actual markers.
Cevabınız için teşekkür ederiz hocam. Bir sonraki sorumuz. Epigenetik ile içindeki bilgi değişmiş olabilecek DNA'dan alınmış olan bilgilerin doğruluğu, stabilite olması açısından ne kadar kesindir?
Can you repeat the question?
Tabii ki. Epigenetik ile içindeki bilgi değişmiş olabilecek DNA'dan alınmış olan bilgilerin doğruluğu, stabilite olması açısından ne kadar kesindir?
So what I understand, if there is a change in the DNA, let's say there is a SNP, and you do an epigenomics, you get some data on it, and then you have a wild type, you do epigenomics, and then they're asking whether the information you get from the non-wild type mutated epigenetic data, how correct it is, because it may not be stable. I mean, if we are talking about a single SNP, I don't think that you would have a problem with the correctness of the information. But I'm also really not understanding the question very well, so I'm sorry. Maybe I'm not answering correctly.
Teşekkür ederiz hocam. Bu son sorumuzdu. Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ederim. Burada sözü Bioinfocongress kurucusu Mehmet Çalıseki'ye bırakmak istiyorum.
Teşekkür ederim. Çok teşekkürler.
Merhabalar. Öncelikle Gamze Hocam, çok teşekkür ederim ben de tekrardan. Aramızda sizi görmekten dolayı çok mutluluk duyduk. Evet.
Duyabiliyor musunuz beni? Çok teşekkür ederim ben de. Beni invite ettiğiniz için çok özür dilerim ilk baştaki teknolojik problemlerden dolayı.
Hiç önemli değil hocam. Yani StreamYard altyapısı daha önce problem yaşatmıyordu hocalarımız ama bu kongrede biraz problemli oldu. Siz kusura bakmayınız asıl. Çok teşekkür ederiz tekrardan.
Hepinize iyi akşamlar diliyorum.
İyi akşamlar hocam.